From the twinkling of distant stars to the vibrant colors of a summer garden, light paints our world with wonder. But light is more than just a source of beauty, it's also a powerful tool for scientific exploration. This is especially true in the realm of microscopy, where light unlocks the secrets of the invisible, revealing the intricate details of life and matter hidden from the naked eye. Imagine peering into a world teeming with organisms too small to see, or marveling at the delicate structures that make up everyday objects, all made possible by the illuminating power of light. Microscopes, like powerful eyes, use light to magnify these tiny objects, allowing us to observe details that would otherwise remain hidden. But how does light, something we often take for granted, possess this remarkable ability to unveil the microscopic world? The answer lies in understanding the fundamental nature of light itself, its properties, and how it interacts with matter. Just as a wave in the ocean carries energy across vast distances, light travels as a wave, carrying energy from its source to our eyes or scientific instruments. This wave-like nature of light is key to its ability to magnify objects in a microscope. By understanding the properties of these light waves, we can begin to unravel the fascinating mechanisms behind microscopy. To grasp how light interacts with a microscope, we must first delve into the fundamental properties of light waves, wavelength, frequency, and amplitude. These properties determine how light behaves, influencing its color, energy, and its interaction with matter. Understanding these characteristics is crucial for comprehending the principles behind microscopy and its ability to reveal the unseen. Imagine the gentle undulations of a wave on a calm lake. The distance between two consecutive crests or troughs of this wave is its wavelength. Similarly, light travels in waves, and the distance between two successive crests of a light wave is its wavelength. This wavelength determines the color of light we perceive. For instance, red light has a longer wavelength, while blue light has a shorter wavelength. Now picture a buoy bobbing up and down as waves pass by. The number of times the buoy completes a full cycle of up and down motion within a specific time period represents the frequency of the waves. Similarly, the frequency of light refers to the number of waves passing a point per second. Just as the frequency of ocean waves determines how often the buoy bobs, the frequency of light waves is directly related to its energy. Higher frequency light carries more energy than lower frequency light. Finally, think about the height of a wave, its crest rising above the still waterline. This height, representing the wave's energy, is analogous to the amplitude of a light wave. In simple terms, the amplitude of light determines its brightness. A brighter light has a larger amplitude, while a dimmer light has a smaller amplitude. These three properties, wavelength, frequency, and amplitude, are intricately linked. They work in concert to define the characteristics of light and its interaction with matter. This interaction is at the heart of how microscopes use light to unveil the hidden details of the microscopic world. When we think of light, we often picture the brilliant white light of the sun. However, this white light is actually a mixture of different colors, each with its own unique wavelength and frequency. This spectrum of colors, revealed when light passes through a prism, holds the key to understanding how microscopes use specific wavelengths to illuminate specimens and reveal their intricate details. As sunlight passes through a prism, it bends, separating into its constituent colors: red, orange, yellow, green, blue, indigo, and violet. This separation occurs because each color travels at a slightly different speed through the prism, causing them to bend at different angles. This phenomenon, known as dispersion, demonstrates that white light is a composite of different wavelengths, each corresponding to a specific color. The range of colors we see, from red to violet, constitutes the visible light spectrum. But this visible spectrum is just a tiny portion of the electromagnetic spectrum, which encompasses a vast range of wavelengths, including radio waves, microwaves, infrared radiation, ultraviolet radiation, X-rays, and gamma rays. In microscopy, the specific wavelengths of light used can significantly impact the image produced. Different wavelengths interact differently with specimens, revealing distinct details and features. For instance, some microscopes utilize ultraviolet, UV light, which has a shorter wavelength than visible light, to achieve higher resolution and visualize finer details. By carefully selecting and manipulating specific wavelengths of light, scientists can enhance contrast, highlight specific structures, and gain deeper insights into the composition and behavior of microscopic specimens. Imagine a straw in a glass of water appearing bent at the water's surface. This everyday observation demonstrates the phenomenon of refraction, the bending of light as it passes from one medium to another, such as from air to water. Refraction is a fundamental property of light, playing a crucial role in how lenses function in microscopes to magnify objects. Light travels at different speeds in different media. When light passes from one medium to another, its speed changes, causing its path to bend. This bending of light, or refraction, depends on the angle at which light strikes the surface and the refractive indices of the two media involved. The refractive index of a material is a measure of how much it slows down light compared to its speed in a vacuum. A higher refractive index indicates that light travels slower in that medium. For example, water has a higher refractive index than air, meaning light travels slower in water than in air. Refraction is the principle behind the functioning of lenses, curved pieces of glass or other transparent materials that can bend light in a controlled manner. Lenses can be either converging or diverging, depending on their shape and how they bend light. Converging lenses, thicker in the middle than at the edges, cause parallel light rays to converge at a single point called the focal point. Conversely, diverging lenses, thinner in the middle, cause parallel light rays to spread out, as if originating from a single point behind the lens. In microscopes, converging lenses are used to focus light onto the specimen and to magnify the image produced. By carefully shaping and arranging these lenses, microscopes manipulate the path of light, bending and focusing it to create magnified images of tiny objects. The magic of magnification in a microscope arises from the interplay between light and lenses, specifically converging lenses. These lenses, thicker in the middle than at the edges, act like tiny light benders, refracting light rays in a way that makes objects appear larger than they actually are. Understanding this dance between light and lenses is key to unlocking the magnifying power of microscopes. Imagine a beam of parallel light rays striking a converging lens. As these rays pass through the lens, they are bent inwards due to refraction. The curvature of the lens ensures that all the rays converge at a single point behind the lens, known as the focal point. The distance between the center of the lens and the focal point is called the focal length. Now consider an object placed in front of the converging lens, at a distance greater than the focal length. Light rays from this object, upon striking the lens, are refracted and converge to form an image on the other side of the lens. This image, formed by the converging light rays, is magnified and inverted. The magnification power of a lens, or how much larger an object appears, depends on the focal length of the lens. A lens with a shorter focal length provides greater magnification than a lens with a longer focal length. This is why microscopes use multiple lenses, including an objective lens with a very short focal length to achieve high magnification. The objective lens, positioned close to the specimen, forms a magnified real image of the object. This real image is then further magnified by the eyepiece lens, which acts as a magnifying glass, producing a final virtual image that we perceive as the magnified object. By carefully controlling the curvature, focal length, and arrangement of lenses, microscope designers optimize the path of light, ensuring that the final image is not only magnified, but also clear, sharp, and free from distortions. While refraction in lenses are crucial for magnification, the way light interacts with the specimen itself is equally important for creating a visible image. Two key phenomena, absorption and diffraction, play vital roles in how we visualize microscopic details. These interactions between light and matter add another layer of complexity to the story of light microscopy. Absorption refers to the process where a material absorbs certain wavelengths of light while transmitting others. When light strikes a specimen, some wavelengths may be absorbed by the specimen's material, while others pass through or are reflected. This selective absorption of wavelengths creates contrast, allowing us to differentiate between different parts of the specimen. For instance, a green leaf appears green because it absorbs most colors in the visible spectrum except for green, which is reflected back to our eyes. In microscopy, stains and dyes are often used to enhance contrast. These stains selectively bind to specific structures within the specimen, absorbing certain wavelengths and increasing their visibility under the microscope. Diffraction, on the other hand, refers to the bending or spreading of light waves as they pass through narrow openings or around obstacles. When light encounters a specimen, it doesn't simply pass through in straight lines. Instead, the light waves interact with the edges and features of the specimen, causing them to bend and spread out. This diffraction of light waves around the edges of the specimen creates interference patterns, where waves either reinforce or cancel each other out. These interference patterns contain information about the size, shape, and structure of the specimen. Microscopes are carefully designed to capture and utilize these diffracted light waves. By analyzing the interference patterns created by the diffracted light, we can gain insights into the fine details and structural organization of the specimen, even at the nanoscale. Light microscopy, through its ingenious use of light and lenses, has revolutionized our understanding of the world around us. By harnessing the properties of light, its wave-like nature, refraction, absorption, and diffraction, microscopes have unlocked a hidden universe, revealing the intricate details of cells, microorganisms, and even atoms. From the earliest microscopes used by pioneers like Antony van Leeuwenhoek to observe microorganisms to the sophisticated modern instruments used in cutting-edge research, light microscopy continues to be an indispensable tool in various fields. In biology, microscopy has been instrumental in understanding the fundamental building blocks of life. It has allowed us to study the structure and function of cells, observe the behavior of microorganisms, and unravel the complexities of biological processes. In medicine, microscopy plays a critical role in diagnosing diseases, analyzing tissue samples, and developing new treatments. It helps identify pathogens, analyze blood samples, and study the effects of drugs on cells. Beyond biology and medicine, light microscopy finds applications in diverse fields such as materials, science, forensics, and environmental science. It is used to analyze the structure of materials, examine evidence from crime scenes, and study microscopic organisms in the environment. As technology advances, so too does the power and versatility of light microscopy. New techniques and innovations are constantly emerging, pushing the boundaries of what we can see and understand about the microscopic world. From super-resolution microscopy, which breaks the diffraction limit of light, to fluorescence microscopy, which uses fluorescent dyes to visualize specific molecules, the future of light microscopy promises even more exciting discoveries and a deeper understanding of the universe at its most fundamental level.